Welcome to Biomed Global. In this video, we are going to talk about sickle cell disease or sickle cell anemia. A recent paper will also be discussed that suggests gene therapy is the hope for the treatment of sickle cell disease. This video is part of the genomic medicine series that talks about the diseases in which human genome are a problem with the genes and their products or the cause of the ailment. A brief introduction about this ailment. Sickle cell anemia is an inherited blood disorder affecting the red blood cells' oxygen-carrying protein hemoglobin. A single molecular change in one of the amino acids of this protein converts the normal protein into the abnormal protein and is the underlying cause of the disease. A person can be a disease carrier or manifests the disease. Why it is like this one? Because all human being receives two copies of each gene, one from mother and the other from father. So if one of the gene is defective and the other one is the normal one, that person will be normal. However, if someone receives both gene defective one, the disease is manifested in the form of sickle cell anemia. This slide is about the molecular change that I discussed. In normal hemoglobin, there is an amino acid, glutamic acid. And in the abnormal hemoglobin, what happened, this glutamic acid in the hemoglobin is converted into valine. So that is the cause of the disease. And here I would like to remind you that all the proteins, they are made up of amino acid, which are almost 20 in nature. As I mentioned, the disease is inherited from parents to the children. So a bit of information about the genetics of this disease. This disease is autosomal recessive in nature. What that means, the gene for this disease are present on the autosome. We all humans have two types of chromosomes. The total number of chromosomes in human being are 23 pair, and among these, one pair define the sex of a person, whereas the rest of the chromosome 22 pairs, they are involved with different characteristics, and we call them as autosomes. So, what it means that autosomal recessive, it means that if the father and the mother both are carrier of the gene, they can have a children which are free of the disease or could be carrier of the disease, and there are chances that their children can also be manifesting the disease if both the defective gene for the hemoglobin, they are being acquired by the child. So that's a bit of information about the genetics of this disease. Here I will discuss how a single molecular change from glutamate to valine in the beta chain of hemoglobin causes the disease. The hemoglobin protein is mainly present in the red blood cell, and if they have hemoglobin with the defective amino acid, these cells lose their elasticity. So here we have the cells which are the normal cells, and in these normal cells, you see the cells which are having a change in the amino acid, and this cell is different in shape than these three normal cells. The difference is due to conversion of glutamate to valine in the hemoglobin of these cells. What happened that these sickle cell, they do not have elasticity and they block blood flow in the circulatory system. Here is the representation. If the cell is a normal, it will just pass through the circulatory system. But if the cell is having a defect, that these cells clog with each other, and this is a typical example of sickle cell anemia, and particularly blood vessel blockage in lungs that lead to chest syndrome, and it can be anywhere in the circulatory system of the human body. Anemia is the major manifestation of sickle cell anemia. However, there are a number of other ailments associated with this disease, like kidney problems, retinal damage, leg sores, spleen damage, gallbladder, liver problems, and so on. Regarding current treatment options for sickle cell disease, there is no cure for the disease. Why there is no cure for the disease? The reason is that disease is genetic in nature. The only way the disease can be cured if the defective gene is replaced with the good one, and that is the gene therapy. However, hematopoietic stem cell transplantation has been helpful in overcoming the disease in certain cases, as the defect is in the red blood cells, which are containing the hemoglobin. So if the stem cells are transplanted, which has the good hemoglobin gene, that can help in overcoming the disease. However, in transplantation, the sibling match hematopoietic stem cell have the highest chance of engraftment and curing the disease. A recent study published in journal Nature in December 2018 suggests that sickle cell disease can be treated with a gene therapy product. So what gene therapy is? In gene therapy, defective genes in human beings are replaced with the good gene. So in this paper, it has been described there is a product called lentiglobin. What that means, it is a viral vector construct containing normal gene for this hemoglobin, defective hemoglobin. So what will happen in this therapy, the defective gene will be replaced with the good one in the hematopoietic stem cell, and it will lead to the production of normal cells, and the people will get cured. Designing and developing a gene therapy treatment protocol is a very complex procedure. It involves the development of gene therapy product, and once it has been developed, it follows with the administration of that product into the human for the treatment purposes. Anyway, for sickle cell anemia, the treatment protocol involve that stem cell with sickle cell gene are ablated in the human being receiving the treatment. Now, new stem cell implanted with correct version of the gene, that is the beta globin gene, and this is supposed to cure the disease. And the treatment protocol takes months, and continued monitoring is required for the gene therapy treatment protocol that is being proposed. Thank you for watching this video. I'm looking forward to your comments and questions regarding this video, and I will appreciate if you subscribe to our channel Biomed Global. Thanks and all the best.